All right, hello biology. Apparently, I didn't get this done, so I'm going to be doing one video for the week again. I apologize. I will get this uploaded as soon as I can. I don't know why I'm telling you this. By the time you see it, it's going to be uploaded. But I'm going to run through everything we did this week. It is Friday. I'm going to run through everything real quick. So, a couple of you have already emailed me about this. Your assignment today, on this day, since this is a 4-day week, was to fill this chart out. Now, for those of you who were stuck at home, you did not get as much help as everybody else did, but the idea was to figure out which words would go into these blanks. So DNA is found where? When it's coiled up, it's called a what? Okay, and then this is made of many of these, and each one of these codes for one what? Stuff like that. Couple that I want to clarify. 6 and 8 are interchangeable. 7 must be correct. And then the pairs 9 and 10 are interchangeable with 11 and 12. And then that will be a little clearer if you actually look at it. If you're confused on this, I can give you a word bank. Well, actually, never mind. Don't worry about that. If you want some help on this, let me know. I don't think you'll need that, but I can give you a word bank if you want. We spent a few minutes on this, and then we switched back to notes. So here were some of the notes about why DNA is considered a unifying molecule. In order for something to be living, it has to have DNA. Now, this is not one of the Gracie facts that we talked about in our first unit. But DNA is part of cells. Cells can't function very well if they don't have DNA in order to allow them to perform specific functions. Okay. This is the first Amoeba Sisters video that is linked in the description of this video on YouTube. There is a second one that we will get to. I think we'll get to that one tomorrow or Friday. I think we might have watched that on Friday. Good Lord, this week has been a blur. I keep referring to today. Anyway. So yeah, after watching that, we kept going a little farther on the notes. Here was a comparison of RNA and DNA. So DNA is 2 strands. DNA is 2 strands. RNA is 1 strand. Notice we have A, T, G, and C, but over here we have A, U, G, and C. RNA does not have thymine. Instead, it uses uracil. And that is one of the fundamental differences between DNA and RNA. So I'll give you a second. I want you to try and fill in this table. Pause this. Fill in the table real quick. Okay. For those of you who are going to, I'm guessing that you have. These are the answers to that. This is why DNA is called deoxyribose nucleic acid, and then ribonucleic acid. That's where DNA and RNA come from. All right. Having watched the Amoeba Sisters video, what would the RNA strand, this is the complementary RNA strand, be for this strand of DNA? Well, what pairs with G? Normally. Well, normally G is going to pair with C. T will pair with A. C is going to be G. And then these are three C's, so they're all three going to be G. A would normally pair with T, but this is RNA, not DNA, so it's going to pair with U, uracil, instead. T would end up with A, G would give us a C, and T, again, would be A. Now let's see if I did that right. C-A-G-G-G-U-A-A-C-A. All right, cool. Now, here is a nice longer strand that I want you to do, but I'm not going to tell you the answer. If you want it, you can email me, and I'll tell you what the answer was. But I want you guys to try it first. And the last thing we did in class on Tuesday was this. There are 7 functions of proteins. This is a throwback to, I think, Unit 3, our macromolecules, where we talked about these functions. All of these functions require proteins, and each of these proteins has to be synthesized first, which is what we're going to start talking about next. Oh, but first, here's the answers to the worksheet that we were doing at the beginning of class. Okay, so Wednesday and Thursday were a little weird. Wednesday, my 3rd hour class did another models lab with this. My 7th hour did not get to do this because and we know it was them because they were fine at the beginning of 7th hour. They damaged several of these kits, and so they got a different worksheet assignment instead. But I went home sick early on Wednesday, and so I was posting that, and I didn't quite get it done before I left. So since 3rd hour did not finish the model activity, and 7th hour I hadn't actually posted their assignment yet, Thursday was time to finish those assignments. So let me go check real quick. Yeah, that was worksheet 5 for both classes. So worksheet 5 for 3rd hour was the DNA replication model set, and worksheet 5 for 7th hour was a reading assignment over DNA replication, followed by several questions out of the text. So that's what we did on Wednesday and Thursday. So then Friday, we continued notes. And if you were not here, you don't have to worry about that. We'll just dive straight in here. Don't need to worry about peanut butter cookies. All right, so protein synthesis is the process of making proteins. Okay, starts with information that is on the DNA, and then we use it to actually, or the cell uses that information to actually create a protein. And protein synthesis is the process of going from DNA to protein. Now, one thing we need to talk about here is the difference between a genotype and a phenotype. Genotype is genetic information. Okay, that is the actual information, the gene that is stored in your DNA. The phenotype is the physical trait that you get as a result of that genotype. So, for example, there is information in your genetic code that determines what kind of protein is going to exist inside of your eye. Okay, and there's some variance on what that protein could be. Some of them give your eye a blue color, others give your eye a bit of a brown color. Okay, and if you've got one of each, then you end up with eyes that might look green, depending on which one is on which chromosome. So then the information that codes for this is going to be the genotype, but the actual color of your eyes is a phenotype. Each gene codes for only one protein, and we say that it is expressed. So your gene, my genes, express as brown hair and gray or blue eyes, depending on what shirt I'm wearing. Okay, and protein synthesis has two primary parts: transcription and translation. And we have a, oh, nope, I'm getting ahead of myself. These are the four things required for protein synthesis to take place. And I'm going to go ahead and mention that a ribosome is primarily composed of rRNA, or ribosomal RNA. You do not have to necessarily remember that rRNA is involved here, but you do need to know that a ribosome is mostly composed of rRNA. So ribonucleic acid, I can't talk today. A single-stranded nucleic acid that contains the sugar ribose, which is why we call it ribonucleic acid. I think I mentioned that already. There are three kinds of RNA. They are messenger RNA, transfer RNA, and ribosomal RNA. Messenger RNA is the strand of RNA that is created by making a copy of the DNA, but in RNA. Okay, transfer RNA transfers amino acids to the mRNA at the ribosome to actually create the protein. So messenger RNA carries the message, transfer RNA transfers amino acids, and ribosomal RNA is one of the main components that makes up the ribosome. And the next three scrolls will reveal those. Oh no, I cut it down to just one. Cool. So there's that. Now, this is the link to the second Amoeba Sisters video, this is the second link to an Amoeba Sisters video where it goes through protein synthesis. And that is where I'm actually going to stop this, and we will pick up here on Monday. If you're in my third hour class, there's going to be a couple slides discrepancy, but that will be on the next video that I do. So you should be good. Have a wonderful night.